Hi, everyone. Welcome to the Spatial Omics Seminar Series 7. And today we are very excited to have Professor Charlotte Scott from University of Gent. And briefly, Dr. Scott received her PhD from University of Glasgow in mucosal immunology. And she did her postdoc at the VIB in the same university, at the University of Gent, as a Marie Curie Intra-European Fellow and Sir Henry Wellcome Postdoctoral Fellow. And after that, she started her faculty job at the University of Gent in 2017, and first as an assistant professor, and now in 2019, she has started her professor career there. And currently her lab is studying mononuclear phagocytes in health and disease. And today's talk will be, of course, about the liver, but you can read more about her previous research as well. And she's been also recently incorporating multiple spatial proteomics and genomics technologies. And hence her recent paper actually says spatial proteogenomics. That's a very original title too. And without, I think, further ado, I'd like to turn over to Dr. Scott. And please, it's all yours.
Great. Thank you very much for the introduction and for the invitation to speak here today. It's been, I've been following these seminars, they've been pretty cool. So I hope I can do it justice. So indeed, what I'm going to talk about is our recent work where we've been trying to generate a spatial proteogenomic atlas of the healthy and obese liver across species. The first thing I want to say is that this work is not done just by my lab. It's a massive collaborative effort. You can see that from the number of authors on the paper, but it's really work that we've done in collaboration with Martin Gilliam's team, also based at the Inflammation Research Centre here in Gent. So when we were starting out with this project, we asked ourselves, what do we really need to be able to make a practical tissue atlas? What would we need to be, to really be able to study these cells moving forward in the future? And so what we decided, and nothing super crazy, is that we would need to have, all of the cells that are present in the liver need to be represented in our atlas. We decided that we didn't just want gene expression profiles, but we also wanted protein expression profiles, and primarily surface protein expression. And the reason that we decided this is because, of course, if you want to go to spatial techniques, confocal microscopy and things like that are still the quickest and easiest and also cheapest way. So if you have the protein that's expressed on the surface of the cells, you can really quite easily potentially move between your map of the single cells versus the spatial arrangement. Of course, I wouldn't be here today if we didn't have the spatial arrangement. And what I will show you is that we've also been looking at this with the protein, but also on the basis of mRNA expression. So I said the first step for us for generating our atlas was that every cell of the liver needed to be represented. And the first thing that we noticed is that the cell isolation technique that you use really matters here. So what we did was we actually compared a range of different digestion techniques to see what cells we were getting out and which ones would then be the best for making sure all cells were present. So what you can see here is a UMAP of an ex vivo digest. So this is a very typical way of digesting a tissue. We take the liver, we take it out of the mouse, we cut it into small pieces, and then we incubate it with enzymes at 37 degrees. We then sort total live cells to get rid of all of the dead cells and debris. And then this is the UMAP that we're left with. And immediately you can see that we have nice populations of immune cells. We have our macrophages, B cells, T cells, and so on, as you can see also represented here. But we pretty much lack all of the structural cells of the liver. So this is not ideal, of course, when you want to generate an atlas. So then we move to another digestion method, and this is an in vivo digest. So the liver is quite unique in that it has a dual blood supply. And so it's very easy actually to perfuse the liver with an enzymatic cocktail and digest the liver then essentially from the inside out. And when we do this, you can see that the map changes quite dramatically. So now we still have immune cells, but you can see if you compare cluster 7, the B cells, for example, that you have far less proportionally when you do this in vivo digestion. We still have our macrophages, but now we start to get out some of the structural cells of the liver. So we have hepatocytes, we have endothelial cells, we have some cholangiocytes, and we have a very small population 3 here, of fibroblasts. Now, as I said, we were interested in studying the liver across both healthy and obese patients and mice. And so we were a little bit worried when we saw this low level of fibroblasts, and this is just in a healthy mouse, because we thought, okay, if we're only getting out this small number of fibroblasts now, what's going to happen when we have a fibrotic liver, as is the case in the obese patients and mice. And so what we also decided to compare was a third technique where we could completely avoid the need for tissue digestion. And this is nuclei sequencing, of course, where you just take the liver out, we snap freeze it, and then we lyse that tissue and homogenize it with a gradient to then purify the nuclei. And you can see when we do this, we again have a completely different picture. So again, very few immune cells, but we still have them, and much bigger population of hepatocytes here in pink. And then you can see we start to have a pretty decent population of the fibroblasts, and you can see this represented here. Of course, then the question becomes, well, which actually represents what we see in vivo? And so to look at this, a postdoc in Martin's lab, Johnny Bonardel, what he did was he performed confocal microscopy, just with some very basic markers to try and understand the relative proportions of the different cell types. So for example, here you can see in red macrophages, so these are stained with F480, and you can see just how abundant these cells are in the liver. Whereas on this side, you can see dendritic cells, so these are stained with CD11C in class 2, and you can see that these are few and far between in the liver, and then mostly seem to be located around portal veins. Similarly, you can stain for things like B cells, so this is with CD19 in red, or T cells with CD3 in red, and you can see that compared to the macrophages, these are indeed relatively sparse cells. And so then what Johnny did was he completely blinded, so he didn't have any of the single cell data, he only had the microscopy data, he counted the different cells and then looked at the relative proportions. And what you can see is if you look at the microscopy, the nuc-seq actually best resembles the in vivo proportions. So if you're looking to create an atlas where you can really have all of the subsets of cells present in the proportions that they're present in vivo, then nuc-seq is the best option, at least in terms of the liver. However, nuc-seq does have some disadvantages as well, and we were able to show that actually we capture less genes per nuclei than we do genes per cell with nuc-seq, as you can see here, and this is just within the macrophage population. But of course, the other big disadvantage of nuclei sequencing is that it's not compatible with the protein methods. And I said before, we were really interested in having the surface protein of the cells, and we did this using the CiteSeq technology. So in the end, what we decided to do was use a combination of the different digestion techniques to build our single cell atlas that we could then take to start localizing the cells, just to be sure that we would have all of the cells present, but that we could also have really in-depth detail about the genes that they express, but also the surface proteins that these cells are expressing. So how did we measure the protein? So as I said, we did this with CiteSeq. This is actually done with the TotalSeq platform from BioLegend. And basically what you have here is antibodies, the same way you would have antibodies for flow cytometry, except these are barcoded instead of having a fluorescent label. So we can actually use panels of up to 200 antibodies, both for the mouse and for the human. And then when you sequence your cells, you can also sequence any antibodies that are bound to your cells. And like that, you get the protein information. To analyze the data, we were analyzing this with the Total BI algorithm from Mir Yosef's lab. So this is a way of looking at both the mRNA and the protein simultaneously. And you can see up here that actually having the CiteSeq data really allows you to better understand your cell population, because this is a Kupffer cell population. This is one of the genes that resident Kupffer cells express, TIMD4. And you can see we have not all of the cells expressing it, and this is just due to the dropout that we see. But with the CiteSeq, you can see you nicely can identify all of the cells. And so this really helps to better identify it and name the different populations that you see. And you can also see this as an example here. So this is clustering of the myeloid cells in the liver if we just cluster on the basis of mRNA, but this is the clustering if we cluster used on mRNA and protein. And you can see that the separation between the clusters becomes much better, allowing these cells that sort of fall in between the different clusters in the mRNA alone UMAP to be really separated when you add in the protein information. And this can also be visualized in terms of the heat maps. As you can see here, these heat maps are a lot cleaner than when we're just clustering on mRNA alone. And that's just because we can really use both bits of information to cluster the cells better. Of course, the most important part about having the protein information is that we can then have these markers to be able to sort the cells for functional studies, or of course, to localize them with confocal microscopy. And just to give you the proof of principle here, what we were able to do is actually take the sequencing data and then load this data into any flow cytometry analysis software. So we use Flojo in the lab. So those of you familiar with flow cytometry will recognize these graphs, and then you can really start to play with the data and gate on it the same way you would gate on normal flow cytometry data, and allow you to develop panels that you can then use to sort the cells and do functional studies. Of course, here I'm showing a manual gating, but with 200 antibodies, of course, it's not the best way to go. And we've also done automated gating strategies using these panels. And here you can just see an example of how we've used this and leveraged it to generate a gating strategy. And this is a gating strategy for the CD45 negative cells of the liver. And so this was quite a surprise to us, but also really shows the power of CiteSeq, because we thought, we're adding a lot of these antibodies, but they're primarily antibodies against CD numbers, and we thought we'd mainly target the immune cells here. But actually, without any prior knowledge really of the CD45 negative cells, we were able to use this large panel of 200 antibodies to develop a full gating strategy, allowing us to isolate the different subsets of these cells. Okay, so now the bit that everybody here is interested in, the spatial side. So once we had our map of the cells and we knew what genes and proteins they were expressing, we needed to assess the question of where the cells were located. And at the time we were doing this, we had two main options, the whole transcriptome approach, but this was not yet at single cell level. So we've done this using the 10x Visium platform, or a targeted approach using probes. And this is of course at single cell resolution or even subcellular resolution, but it's targeted so you don't have the whole transcriptome. And for us, the choice really depends on the specific question and also how well you know the tissue. And because we wanted to have a completely unbiased atlas of the liver, we decided to start with an unbiased whole transcriptome approach and then go to single cell resolution with the genes that we had found to be spatially resolved on the different cell subsets, using both our single cell map coupled with the Visium approach. So as I said, we used the whole transcriptome approach from Visium, so probably everybody here knows how this works, but just in case, you have these slides that have these capture areas, and on these capture areas, you have the different spots. And these spots then have a specific barcode, so you can map back the location. They also have the UMI and the poly-DT. And then what you do is you just place your tissue slice, you lie this on the slide, you lyse the tissue, and the mRNA will then come out and bind to these spots. And then you can map back exactly where the mRNA came from. One note about the liver. So as I said, the Visium is not single cell resolution, but the liver is actually quite useful for Visium analysis on the basis that it has a lot of these repeating structural units. So you have many central veins here shown in yellow and a lot of portal veins. And we know from previous studies that the hepatocytes around the portal vein and the central vein have very distinct gene expression profiles. And so like this, you can actually start to get some idea of where you are in the tissue by comparing with these portal and central vein associated genes. And if you just take another image as an example here, so here we have a portal and a central vein, and here you can see the scale bar is about 100 microns, so about 2 Visium spots. Of course, if you then go further with the Visium, we know that you'll have areas like this, but you can clearly see that we'll be able to delineate the differences between a central and a portal vein using this method. And so this is some of the data that we've got from the Visium analysis. This is just a healthy liver tissue, and here you can just see the clusters that we were able to identify overlaid back onto the tissue. And you can already start to see some of these patterns appearing with these repeating units. Of course, you can also look at these clusters in a UMAP. But what was really interesting is in collaboration with Wouter Salens and Bart de Planke, we were able to use a novel algorithm that is coming soon on GitHub and bioRxiv called Latenta, which allows us to actually predict what cells are in what location. And the way we did this was to first order the spots on the basis of their expression of known portal and central zonation genes. You can see you have the portal region down here and the central region up here. We also had some liver capsule, which you can see located here. And then of course you have the areas in between the periportal and the mid zones. And then, okay, so we've ordered the spots, and then you can look at this ordering, and now you can really start to see this central portal structure of the liver appearing. And then what we did is we took the gene expression profiles we have from our single cell data set, and then using the Latenta algorithm, we asked, well, what is the likelihood of finding this expression pattern in a specific region of the tissue? And like that, we could start to localize the different cells. So for example, if you take the conventional dendritic cells here, so we predicted that we're most likely to find the signature for these cells in the portal region. Similarly, if you look at the Kupffer cells shown here in blue, these are primarily periportally located. There's not many really at the portal vein, and there's not really very many at the central vein. You can also look at this, of course, on the UMAP. So again, you can see that the Kupffer cells are primarily found periportally, whereas dendritic cells are more located in the portal region. Of course, then you want to go on and validate these predictions. And so we wanted to move to single cell resolution. And as I said before, we ideally wanted to do this with confocal microscopy because it's relatively cheap and efficient. But the one problem that we had, even though we had the site-seq data, is that antibodies that work by flow cytometry, for example, or by site-seq, they're not necessarily going to work on the fixed tissues. So the question that we had was, how do we best screen this panel of 200 antibodies for the ones that are going to work best in tissue sections and fixed tissue sections? And so what we decided to do actually was adapt the Visium protocols, and we did this with the help of, or in collaboration actually with Marlon Sokias at 10x Genomics, to combine the Visium with the site-seq to be able to see directly on the tissue which site-seq antibodies are binding and which ones would be useful to delineate different spatial profiles. The one thing that I have to mention here, of course, is the Visium system as it's set up is designed for RNA. When you are manipulating the tissue to get the best RNA signals, you're not doing much that's helpful for the surface protein. And if you want to preserve the surface protein, then you're going to have issues with the mRNA. We were able to find a sort of middle ground where we could get signals from both the RNA and the protein, but you can see that compared to the classic Visium shown here, we do have less genes per spot, but we still have enough. We can still look at the mRNA, but it is less than what you have with classical Visium alone. So it's just something to bear in mind. But it does work, it works quite well. So for example, this is the mRNA profile on the Visium for CLEC4F, which is our favorite Kupffer cell gene in the mouse. And here you can see the protein, and you can see that these signals are fairly similar and overlapping. So it's really working quite well. So same idea here with CD55. This is also a gene that is expressed by Kupffer cells. It's pretty low in mRNA level, but we know that it works really well by flow cytometry, and it came out really nice in the site-seq. And here you can see spatially it's also working. So by having all of these bits of information, you can then decide that actually CD55 might become one of the top candidate proteins for downstream functional analysis and spatial analysis. Similarly, we were able to identify antibodies that are really spatially located. For example, Ly6A and Ly6E, they really come out as being portally regulated in our single cell map, and also you find them around the portal zones on our Visium site-seq data. Just as we analyze the mRNA, you can also do the same with the protein and then look, where are the protein signals coming up? And for example, if you take in blue here, these are our Kupffer cell markers. You can see again a bias towards the periportal region, but we were also able to identify antibodies that are really located with a distinct spatial orientation. So for example, you have SCA1 and CD105 here, which are present at opposite sides of the spectrum. And so by having all of this information together, we were then able to go to a sort of multiplexing protein situation. And for this, we were using the Miltenyi Biotech Maxima technology, which we were able to get early access to through the VIP TechWatch program. And this is a technology that allows you to combine up to about 100, I think they can even go to about 200 now, different antibodies simultaneously. It's a very cool system in which the antibodies are made with a cleavable tag. So you don't need to bleach the tissue every time with the sequential antibodies, rather you just cleave off the tag and then add on the next ones with the tag, and then you can measure the fluorescence like that. So this is an example where we did it with 60-plex protein in the mouse liver, and you can now nicely see that we can really localize the different cells. The Kupffer cells are shown here in blue, you have the L-sex in orange, T cells, monocytes, and so on. You can really now start to identify them and determine where they are compared to the portal and the central veins, which are marked here by cholangiocytes and hepatocytes expressing glow, respectively. Now, this is kind of a good news story so far. So of course, it's very easy to find antibodies that allow you to tell the difference between a macrophage and an endothelial cell, or a macrophage and a T cell, but it becomes a little bit more complex when you really want to study the location of different subsets of the same type of cells. So for example, we're really interested in different subsets of macrophages. And so sometimes there just is no antibody available that's going to allow you to do this. And so when that's the case, you obviously need to turn to targeted mRNA approaches. And for this, we use the Resolve Biosciences molecular cartography platform, which is a 100-plex targeted fish-like approach. And to decide on which genes we would use for this, we took the top DE genes from our single cell map, but we also combined this with the differentially expressed genes that were differentially expressed at a spatial level on our Visium analysis. And like this, we were able to come up with a list of 100 genes. And just to give you an idea that this is working, these are just some of our zonation genes that I was talking about earlier that allow you to distinguish between the different regions of the tissue. So you have central veins shown in red here and the portal veins in dark blue, and then everything in between. So you can see that the molecular cartography approach really works very nicely and allows you to completely recapitulate the zonation within the liver. Just to give you a bit more data to convince you that this resolve system works beautifully. So this is some confocal analysis that we have where we're looking at CLEC-RF for Kupffer cells in yellow, and F480 in red, of course, for macrophages. You have EBCAM in cyan, and the endothelial cells is the CD31 in white. And you can see you can have pretty much the exact same image with the resolve when you're using these exact same probes now, looking at the same genes encoding the proteins that we use for the confocal. Of course, it's a slightly different piece of tissue, so the image isn't exactly the same. And here we're lucky in that the genes that we've chosen, they're highly expressed, so we get a lot of reads per cell, and then you can really start to see a sort of cell-like structures appearing. But as I said, this really convinced us that the resolve was the way to go, that this would work really nicely. Just to give you another example with two highly expressed proteins, you can even recapitulate cell-cell interactions using the resolve data set. So here you have an interaction between Kupffer cells in red and stellate cells shown here in green with Desmond staining with the confocal. And you can see here you have the same interaction of these cells also just on the basis of mRNA expression, which is really cool. And so what we were able to do then is go all the way through from having the single cell map to identifying the top DE genes to then identifying the best probes to use for the resolve. And like that, we've been able to localize all of the different cells of the liver. And this is just an example here showing the different endothelial cells where we were able to find subsets that are specific to the central vein versus subsets that are specific to the portal triad. One thing that we also wanted to do is take it one step further. And now I showed you the mRNA profiles when we have really highly expressed genes where you can almost see the outline of the cell. But if you have more lowly expressed genes, this becomes a little bit more difficult. So for example, here, what you can see is mRNA for some of the DC genes, so FLT3, XCR1, and so on. And these are expressed, but they're relatively lowly expressed. They're also quite small cells. So it becomes a little bit difficult to know where one cell ends and one cell begins. And so what we wanted to do, and really what our aim is moving forward now, is to be able to combine mRNA with surface protein to be able to really see the structure of the cell and then be able to quantify the mRNA that's in there. And so we've been able to do this successfully up until now, and this is using RNA scope protocol with conventional confocal microscopy. And so we're now looking at ways that we can do this, combining the resolve mRNA system with some confocal staining to really allow us to segment the cells and study the mRNA locations in a slightly more quantitative way than what we've been doing up until now. Of course, the very cool thing, so while I've shown you all the data from mouse so far, it's not just compatible with the mouse, all of these techniques are compatible across species. And so we were also able to do all of these techniques also in the human liver. And this was done in collaboration with a number of clinicians, so Hans van Vleerbrugge, Lindsay Vitesse, and Bart van der Voort at University Hospital in Gent, where we were able to get 19 different human liver samples and then perform the same analysis, different types of digest with CiteSeq and the resolve and Maxima technology to really allow us to localize the cells. And so what I hope I've convinced you of so far is that by using these different multi-omics techniques together, you can actually go from having not just a map of what cells are present in the tissue, but you can also start to understand where they are. And moreover, with the antibody information, you can go on to develop microscopy panels, flow cytometry panels, and then really go on to look at the biology of the cells. So in the next part of the talk, what I want to discuss with you is how we use then all of this to look at some of the biology of the liver. And so the question that I'm going to talk to you about today is hepatic macrophage heterogeneity, which is a subject that's really close to our heart. We were really interested in understanding once and for all what subsets are present in the healthy and obese liver, where they are, and why they are in those locations. So just as a brief introduction, when we started this project, we knew that we had obviously resident Kupffer cells, so the majority population of macrophages in the liver, and actually one of the biggest populations of macrophages in the whole body. But we also know that you have a population of macrophages called capsule macrophages, which reside in the liver capsule, which you can think of a little bit like the skin of an apple. This is the case in homeostasis. In inflammation, we knew that you also had additional macrophage populations recruited, but exactly what markers we should use to delineate them was not very clear. We also know that there are a couple of different subsets of these, because not only can a macrophage come in and become something that looks very different from a Kupffer cell, a recruited macrophage can also start to take on a Kupffer cell profile, and with time, look more and more like a resident Kupffer cell. while we had a bit of an idea what was going on in the mouse, we had much more limited data on the human macrophage side of things. And so while we know that there are cells present and potentially other recruited macrophages, you can see here that the markers that people were using to delineate them were not that different between the subsets, and often relied on expression levels. So for example, CD32 high versus intermediate, rather than positive and negative. And that of course makes it a little bit difficult to know lab per lab, are we really looking at the same cells? And so what we've done here is we took our whole mouse atlas that we had, and we zoomed in on the myeloid cells, and then we looked in much more detail at what populations that we had. And here you can see the UMAP that we get for the mouse liver, and you can see you can have this huge population of Cooper cells, but we also had populations shown here and here of macrophages that we were not really able to immediately identify. And when we looked at the differentially expressed genes for these populations, we thought that this cluster 7 over here express a number of genes that look like they may be the capsule macrophages of the liver, based on this paper from Sierra et al. So including things like CX3CR1 and CD207. If we looked at this cluster 8, which is a very small cluster, but here nonetheless, it really resembled what we had previously found in the fatty liver, so a population that we had called lipid-associated macrophages at this time, expressing things like GPNMB, SPP1, TREM2, and CD9. And so we wanted to look into this in more detail and really understand where these cells were located. Unfortunately, when we looked at the differentially expressed proteins that we had from the site-seq, while we had really good markers for some of the cell types, including the Cooper cells, we could not really identify any good proteins that would allow us to separate these populations. And so for the site-seq, while we can identify a nice gating strategy to get out the different monocyte subsets and the dendritic cell subsets and the Cooper cells, we really couldn't separate these non-Cooper cell macrophages. As you can see here, they just fall back into one cluster. Just as a slight side note, so this is the site-seq data shown in FlowJo, but we're also able to take this exact gating strategy, but then use fluorescently labeled antibodies and really confirm it. So this is just the exact same gating strategy, but now with real flow cytometry, just showing how powerful site-seq really is for identifying the different subsets. And again, here you can see we could find these non-Cooper cells. They're relatively rare in the liver, but they are there. And again, we didn't have any markers that would allow us to separate these in more detail. And this is just a validation of that. So once we had shown that we could use the same gating strategy, we sorted the cells and then looked by qPCR at the top differentially expressed genes, just to show that, you know, what we are calling a CDC2 or a Cooper cell really is expressing the markers that we find in those clusters on our single cell map. Sorry. So without any proteins, we went back to the genes to see if we could separate these clusters 7 and 8. And what we identified as CD207, as I already mentioned, is a marker of the capsule macrophages, and GPNMB is a potential marker of the lipid-associated macrophages. CD207 was in our site-seq panel, but it doesn't work very well because it works better as an intracellular protein, and of course our site-seq was extracellular. And so what we did is some intracellular staining for CD207, and indeed we can split these non-Cooper cell macrophages into 2 different populations. What was interesting is when we only isolated the capsule of the liver, instead of taking the whole liver tissue, you could see that we had an enrichment of the CD207 positive macrophages in the capsule tissue, which made us indeed think that what we were looking at here were the capsule macrophages. And indeed, we could find a nice population of capsule macrophages with normal convolvulus microscopy. But unfortunately here, CD207 didn't work to allow us to confirm this. And so what we did is we turned to the resolve approach again, and now we're able to use CD207, but also, you know, a range of other macrophage markers that came out of our single cell analysis to be sure of our identification. And indeed, what you can see here are these CD207, so purple macrophages present at the liver capsule, confirming that the CD207 positive population are indeed capsule macrophages. But what was really interesting to us is that while we were able to find these capsule macrophages, we also found a number of non-Cooper cell macrophages present around the different veins. So here you can see these cells are green and not red. So the red and green are the Cooper cells, but the ones that are green only are these non-Cooper cell macrophages. And these are present both at the central vein and the portal vein. Sorry. We then went on to look and see what genes they were expressing. And you can see quite nicely that we were also able to find CD207 positive macrophages also at the central vein, less so at the portal vein, but primarily at the central vein. So by adding this level of spatial information, we were able to understand that this signature that had previously only been associated with capsule macrophages actually identifies the population of macrophages that are present both at the capsule and at the central vein. On the other side of the coin, if we look at expression of GPNMB and these other lipid-associated macrophage genes, we see that these are primarily located around the central vein and particularly in close association with bile ducts. And so we were able to, really define this lamb-like population in the healthy liver, and we decided to call them bile duct lambs based on their location. So this is really an example of how just having spatial information allows you to go one step further in identifying the different populations and naming them. So now we have our capsule and central vein max and our bile duct lambs. The really cool thing was that the lambs were also present in the human liver. So here you can see our mouse liver signature of the lambs, and we were able to map that in our human liver samples, and you find two clusters that have a very similar profile. And you can see that also here on the single cell map. So you can see it's these two clusters here that seem to resemble the murine lambs. What was really cool is, as I said, we previously identified these cells in the obese murine liver, and we did also notice that in the couple of patients that we had, it's not very many, that had a lot of steatosis in the liver, the lambs also seem to be increased in those patients. So it seems to correlate there. We wanted to understand, are the lambs also present around the bile duct in the human? And so we went back to the Visium system to see where they were. Here we're looking at a pool of patient livers. So it's actually 4 different patients, 2 of which had were obese. So they had more than 10% steatosis in the liver, and 2 of them were lean and relatively healthy patients that had less than 10% steatosis in the liver. And they're shown in purple for the lean patients versus the orange color for the obese patients. And you can see immediately that the Visium clusters completely separate on the basis of the presence of lipid or not. Just showing you how much the gene expression profile changes just by having the presence of some fat in the liver. We then took the same latenta approach that I explained earlier to understand, to try and estimate where we can expect to find these lipid-associated macrophages in the liver. And what you can see here is that very similar to in the mouse, in the healthy patients, we see them primarily around the portal vein, which would be consistent with the bile duct location. Whereas in the obese patients, these are primarily located around the central vein. Sorry. Here you can see this a little bit clearer. So here you can have the different clusters that we identified in the Visium analysis mapped back onto the tissue. This is a healthy patient. This is an obese patient. And immediately you can see that these clusters are completely distinct, as I showed you in the previous slide. But what you can really see here is that you have specific clusters that are really, this blue cluster, for example, that is really located where you have the steatosis. So this presence of steatosis really changes the presence of the genes that are expressed in these regions. And if we then look at the zonation patterns to look at portal and central zonation, you can see that indeed this pattern of steatosis here that was marked by the blue cluster perfectly correlates with the presence of the central vein associated genes, suggesting that indeed this lipid is present at the central vein location, and suggesting that the lambs in the obese patients are potentially moving away from the portal zone, but to be found present in these regions of steatosis. So that was really interesting to us. And what we think is happening is not so much that the ones at the portal vein migrate, but rather that you have recruitment of monocytes to the liver, which would then be recruited to these zones of steatosis, and it would be something in that zone that would then tell them to differentiate into a lipid-associated macrophage. We wanted to validate this in the obese mouse setting. So this is some confocal microscopy from a PhD student in my lab, Anna Lane, and what she was able to show is that indeed the presence of these LAMs, these SPP1 positive macrophages, correlates with the presence of steatosis, but also fibrosis. And we were also able to see this in terms of our Visium analysis, whereby the LAMs shift more towards the central region when you have a mouse fed a Western diet. So you have obesity. And so with all of this together, what we were able to sort of show was that in homeostasis, we have actually 3 distinct subsets of macrophages. You have your resident Cooper cells, you have your capsule and central vein macrophages, and then you also have these LAMs associated with bile ducts. In human, we were also able to find them, and we were able to identify better markers for these cells. I didn't go into that in detail today, but for example, EC4 and FOLR2 are 2 really good protein markers of resident Cooper cells in the human. And just like in the mouse, SPP1 and GPNMB are good markers for the LAMs in the healthy human liver. In the context of inflammation, and in this case, we're talking about obesity and fatty liver disease, we know that we have a population of LAMs that are also recruited, and then these are recruited to zones where you have steatosis and fibrosis. And then you also have a population of cells that are going to become what we call monocyte-derived Kupffer cells. So they start to look very much like the Kupffer cells, and they take on all of the same markers that we see in the resident Kupffer cells, except initially they do not express TIM4 or at protein level in the mouse or at mRNA level in the human. So having all of this information, we're of course then wanted to ask the question, well, what is driving the identity of these different populations? Why do we have all of these different populations of macrophages? And given the fact that we now know from our spatial data that they're located in different regions of the liver, is location and the local environment of these cells driving these different expression patterns? And so what we decided to do is look firstly at the Kupffer cells, so that we know exactly where these cells are located. We know that they're in contact with hepatocytes, with stellate cells, and with endothelial cells. And so we looked at what was conserved between mouse and human in terms of the Kupffer cell profile. And then we looked at using NicheNet at the ligand receptor interactions between Kupffer cells and those different cells that I just mentioned. So the stellate cells, the endothelial cells, and the hepatocytes that could be inducing this conserved Kupffer cell phenotype. And we were able to identify an interaction between hepatic stellate cells, which express BMP9 and BMP10, which is BMP9 is encoded by GDF2, which binds to a receptor on the surface of the Kupffer cells called ACVRL1 or ALK1. And here you can just see the expression of ALK1 in the human Kupffer cells here at the top. And this is the expression of BMP9 and BMP10 in the stellate cells, again in the human stellate cells. What was interesting is, we identified this as being conserved in mouse and human, but we actually profiled 7 different species, and it was conserved. This expression pattern was conserved in all 7 species. So we got really interested in studying this to see how important it would actually be in regulating the Kupffer cell phenotype. And so to look at this, what we did is we took ALK1 flox mice and crossed them to a CD64 crease, CD64 being encoded by the Fc gamma receptor 1 gene. And you can see in a normal liver, these are our Kupffer cells, they're VC4 TIM4 double positive. But in these mice that we lacked this ALK1 receptor only on the Kupffer cells, you can see immediately from the flow plots that we had a reduction in the Kupffer cells. They lose expression of TIM4, and we have this huge recruitment of these VC4 negative macrophages. And this you can just see here enumerated. So we have an almost complete loss of our VC4 positive Kupffer cells and this recruitment of these VC4 negative macrophages. It's not only loss of VC4 as one marker, but we see that they change their expression of a number of Kupffer cell associated genes. So they reduce their expression of CLEC4F, they reduce their expression of CLEC2, CD38, and CD14, just to give a few examples. We then went to confocal microscopy to see what was happening in these mice. And you can see here, this is staining by a postdoc in Marchan's lab, Camille Wagner, and she's looking for Kupffer cells. So Kupffer cells should be VC4 and F480 double positive, as you can see here. But in these mice where we've removed just the ALK1 receptor and only from the macrophages, you can see we have an almost complete lack of Kupffer cells in these animals and just some of these VC4 negative macrophages remaining. So by going through all of these different steps, we now have a model where we can completely see the importance of this pairing of a Kupffer cell with a stellate cell. They interact via BMP9 and BMP10 with ALK1. And if you can just have all of this information, you can then block one single receptor and have a mouse that's almost completely devoid of Kupffer cells because they do not have the ability to get these signals and develop as required into a genuine Kupffer cell. We were also interested in doing the same for the lipid-associated macrophages, and we tried to do the exact same analysis. And here we were really interested in signals that would then be conserved both at the portal vein and around the bile ducts in the healthy individuals, but also in the regions of steatosis in the obese individuals. And unfortunately, unlike the Kupffer cells, we didn't really find any very strong signals in these 2 locations conserved across species. So then we started to think, okay, what is special then about these locations where we find LAMs? And we started to think maybe it was a local metabolite that was driving this. And one thing that we were able to find, so this is work by a PhD student in the lab, Federico, was that if you just culture monocytes with acetylated LDL, so lipid, you can actually induce the LAM phenotype in vitro. So you can increase expression of GPNMB, CD9, and PLA2G7. And what's really cool is that in certain models, so this is looking at a paracetamol overdose model, you also have an increase in lipid in these regions where you also start to see lamb-like cells in this model. And so just to summarize everything I've shown you today, by having this spatial resolution, you get this whole new level of information, which allows you to first better define the different cell types and allowed us to identify, for example, the bile duct lambs in the central vein and capsule macrophages. But it also allows the identification of which cells are interacting with which other cells and how these local interactions may be driving identity. So we were able to do this for the Cooper cells, where we've shown that ALK1 and BMP9 and 10 are crucial for Cooper cell development and maintenance. For the lambs, we were also able to show that this isn't really the case, and rather it seems to be the lipid and local metabolites that may be driving this lamb signature. And of course, this is something we're investigating further. The last thing that I want to mention is that all of this data is available online. So if you go to livercellatlas.org, you can look at all of our UMAPs. You can search for any gene you're interested in. The Visium data is also there for download, the Resolve data, the Maxima data. So if there's any question that you have, you want to look at any of this data, please go ahead and use it. And if there's something that you don't find immediately on the website, just drop us an email and we'll be happy to help you. I think this is this is the power of all of this high level data, is that we can really share and address a number of different questions from it. So we're more than happy to collaborate with anyone who's interested. And so with that, I just want to thank everybody that's involved. As I said, this is a massive collaborative effort between my team and Martin's team, and this is almost everybody shown here. I also want to thank the clinicians for the human data, all of our core facilities, especially TechWatch and the Single Cell Accelerator program, which allowed us to push a lot of these technologies to the max very early on. The Latenta analysis is done by Wouter Salons and Bart de Planke. We also collaborate a lot with Ivan Size and Robin Brouwers for the NicheNet analysis, and of course the funders. And with that, I will be happy to take any questions.
Thank you, Charlotte. Just so impressive. Such a kind of massive, complete data set. It's a very valuable resource. So I think already 2 questions in the chat box. So do you want me to read for you, or you can?
I can read it, that's okay. Yeah.
Okay, you can look it up.
So yeah, the first question is whether we plan to investigate the human tissues from patients who progress from NAFL to cirrhosis. So this is something we would love to do. We don't have the cohorts at the moment in Belgium. So if you have, or you know anyone with these cohorts, we're more than, we'd be very happy to look into this. It's something that we're trying to set up a collaborative effort across Belgium to start looking at. So indeed, that's where we want to go next. Okay, the second one, 2 questions. Yeah, so the stellate cells and the Cooper cells are normally on the 2 different sides of the endothelium. How do you think they interact? So this is really cool. The endothelial cells, that liver sinusoidal endothelial cells, are fenestrated. And we've actually been able to show in previous studies, it's actually worked from Martin Gilliam's lab, that the Cooper cells, while they reside with most of their body in the endothelial, in the blood vessels, they actually extend their arms and their out through the fenestrations where they come into contact with the stellate cells. And Martin has beautiful images where you can really see the stellate cells and the Cooper cells interacting. And so that's how we think this interaction is occurring. I actually think it's something that's particularly interesting in the context of fibrosis. When you can lose these fenestrations, we actually know that you also see a reduction in Cooper cells in those models and in those patients. So I wonder if it's, you're losing the Cooper cells because you're actually blocking their ability to interact with the stellate cells. The second question. Yeah, so it's about where the lambs are located in the obese liver between mouse and human. So the regions where you have steatosis in our mouse model is different to the regions where you have steatosis in the human. And so what we see is that the lambs, they really seem to locate in these regions where we have steatosis. And that depends patient to patient and also diet model that we use. The other thing that we're looking into, which is something that I didn't stress in the talk, is potentially there may be some dysregulation of some of the known zonation genes when you have a fatty liver disease. So genes that we typically associated with the central vein region, when we look at those, how they're located in the the resolved data, there may be some skewing. So instead of being solely present at the central vein, they seem to be present throughout the tissue. And so we're wondering if actually the presence, some of these zonation genes, they're there because of hepatocyte metabolism, and the hepatocytes around the central vein are known to function in lipid metabolism. So we wonder if you don't get lipid present in other places of the liver because of the high fat diet, that you wouldn't actually induce expression of some of these cells and some of these genes. And so we need to be a little bit careful with what we call zonation genes in obese and inflamed settings. And that's something that we're looking into in more detail now.
Yeah, I think to follow up on this question, so at a very high level, you think those kind of distinctive phenotype, for example, that's largely kind of regulated by just a metabolic environment as kind of reversible phenotype, or that's something rare like a cell type versus cell state. The cells kind of stuck in that cell type for long enough time to really drive the disease or something. And or have you looked at other layers of the mechanism?
Yeah, so it's a super interesting question, and it's something that we really want to do, but unfortunately we still lack the tools to really fate map these cells and see what would happen if we would, put the mice back on normal chow. Do they stay? Do they change into something else? We're generating the tools at the moment, so hopefully in a couple of years I'll be able to answer those questions. It's not a Cooper cell. What we know for sure is it's definitely not a Cooper cell that changes its phenotype in fatty liver disease. It's a recruited monocyte that comes in that takes on this profile. Whether it can then go on to become a Cooper cell, because we do have these monocyte derived Cooper cells also appearing, that we don't know, and that's what we want to study when we have the fate mapping tools. Time will tell.
Also some technical question, I think, in your Visium data from 4 different patients, multiple sections, how did you normalize? How did you kind of do batch correction in this case? Sometimes we're struggling.
It wasn't easy, that's for sure. I would have to double check exactly how we did it in the end. But for that, we really collaborated a lot with Bart de Planke and Wouter Salens. They really helped us a lot with the analysis and how to do this. We did look at the patients individually before pooling them to really make sure that we weren't having issues with, something that was coming up just from pooling the patients, and that definitely didn't seem to be the case. And there was also a lot of comparison with the 19 different patients that we had in the single cell analysis. But I have to say, I don't remember exactly what batch correction method we used, but I can definitely put you in touch with the people who will have the answers for them.
Wonderful. Maybe one question from here as we wait for others. The RNA protein data sets in identification of cells and their roles, right? It's pretty impressive, but one question is, in terms of the validations, right? I see that the RNA counts are really, really low. It's only like a few dots per area, right? And some of these assays that you showed require protease digestion that might actually change the biology totally compared to protein staining, right? And protease is, it's actually opposite of the other one. So what would you think about the validations needed for the future integration, as well as, how can we really take this to a more, robust approach for proteogenomics?
So we spent a lot of time optimizing the protocol to allow us to be able to do the two things simultaneously. And actually one of the steps that we removed is the protease. So we don't do any protease with the RNA scope and confocal, because it just, you just lose the protein signal basically. That's one side of the coin. The other thing is, I will say that if you do mRNA alone for those genes and don't combine it with the protein, the mRNA signal is much higher. So we're clearly not getting all of the information. And I think that maybe one way that we can validate it is, now we're doing everything on one slice, which I think is important, but you can also have sequential slices where you can do mRNA alone versus protein alone, and then really make sure that we're not skewing what we see on a very similar piece of tissue. And that's actually how we did it to double check everything. Yeah, long term, I don't know exactly where it's all going to go, but I really think that having the surface of the cells is going to be crucial for any sort of segmentation. You know, we see it, the nuclei alone, it's not sufficient to segment the cells, especially in a tissue where, you know, all the cells are layered on top of each other, especially in a relatively thick section. So yeah, this is why we think it's where we need to go to really be able to do any sort of quantitation of the RNA analysis. But there are caveats in that the mRNA is lower. So I think developing the protocols that will allow us to keep everything to the max is the key there. But
yeah, totally. Because the RNA data, when I look at it, you know, it's very sparse. Looks like that there's still a hidden, like this dark matter at the background, right? And that actually would be important. And then you also show these zonation patterns. Are they done in 2 dimensions, or they are done in a like, because you have these kind of plots, right? Are they like a just line scan, or are they like an integral of an area? Like how would you look at the, because it's a 3-dimensional thing, right? Based on
yeah. No, it's just a line plot. We're just it's a pretty simple representation where we're just, you know, putting all of the cells along an axis from portal to central on the basis of the genes that they express, and then predicting where we think the signatures of the different cell types will be found. But indeed, yeah, that's the next thing, of course, is to build that onto the tissue and be able to look in 3 dimensions. And yeah, it's all still to come, I think.
Sure, but this is beautiful. I mean, all these zonation patterns, and that's really physiological, you know, regions, and pretty interesting. Anyone else? I think we have currently 9 people.
So in this case, actually, I think a 2D works very well, right? Because the portal vein, central vein are coming, for example, there's a piece of paper that's coming from top to bottom, right?
Yes.
So that's kind of like a kind of quasi 2D
approximation. Totally.
Yeah.
Totally. Yeah, that's beautiful. I was just curious. It's a 3D thing and how that scales up.
Yeah, no, unfortunately for now, it's just 2D, but it's definitely something that I know Wouter and others, the bioinformaticians, are interested in, and it's beyond my realm of abilities, but I defer to them on all of this.
Sure.
Yeah.
Thank you so much.
So maybe you mentioned, I forgot, I didn't, I probably missed, how many proteins you look at in your CySeq?
In the CySeq, on the cells, on the dissociated cells, we have panels of about 200 antibodies. For the Visium, we did 100 because we had issues with having enough antibodies and the wrong dilution. Yeah. So for the single cell, it's with panels of, it was, I think, 150 something for the mouse and 189 for the human.
Okay, yeah, that's the biology, the cocktail they provide.
Yeah, we basically bought every antibody from BioLegend at the beginning. So yeah, we were really lucky there with the single cell programs at VIB. They, you know, a lot of PIs came together, and we were able to get a lot of the antibodies in and use them. It was also in collaboration with Janssen, so it was really, really nice. Yeah,
so I think you should you're showing a UMAP, the clustering from the transcriptome data, and also then you use the proteins to either, I don't know how you computationally, you either embed or integrate and perform clustering. Have you done a clustering of the proteins owner? Since you have like 200 proteins, you may get a very nice clusters from just the proteins.
Yeah, we have done the proteins alone. You do get nice clusters from most cells, but then there are some, for example, we wouldn't have seen the lambs because we didn't have any good antibodies to really separate those from other cells. So it definitely works with protein alone, but unfortunately we just don't have, even with 200, we don't have enough to separate all of them. So that's,
wonderful. And one interesting thing also your finding was the dissociation protocols, right? And how that affects the cell populations. I think that's really probably a very important key topic for many organs that we study, right? Or the biology that we study. And currently, you know, we probably just study it by dissociating using enzymes or whatnot, right? Or mechanical But there was also another paper in the Schmetz earlier actually showing that the dissociation actually changes gene expression, right? Not only population. So I think that actually would be very interesting. And do you have any vision for where we are heading for different organs? Let's say you worked on gut, right? Are these scalable, right? What do you think?
Yeah, so I think it's a really important question. You know, how much are we changing the gene expression profile by, you know, digesting at 37 degrees for half an hour or you know, whatever it is for the different tissues? We definitely do see some, you know, activation digestion genes associated in our digestion protocols that we don't see in the nuc-seq. So actually having both is quite a good comparison. But the nuc-seq alone, I don't think is ever going to be sufficient to take over from the cells, because there are a number of genes that are not associated with dissociation that we just don't capture in the nuclei sequencing. So I think you really need both. I think what we're trying to do at the moment is investigate the use of these cold enzymes that you can do the digestions on ice to see how much that would, you know, prevent some of these gene expression changes. And I know some other people in the institute are also, you know, trying to block transcription during the digestion as well. So I think it's these sort of approaches that we're going to have to use to really be able to get to the bottom of it and at least figure out what part of the signature is associated with just dissociation. But I think that's also where the spatial comes in, because yeah, you're not going to have those genes in a spatial data set if you're not digesting the tissue. So like that, you can already rule out quite a few. So already the addition of the spatial helps a lot in that.
Yeah.
Totally. But of course, in the spatial, it's important where you're looking at too, right? Because the biology here and there might be two different things. And
yeah, this is what we struggle with the most at the moment. Yeah, because the cell, especially the Cooper cells, they're big cells, they're reaching across things, and yeah, you're only capturing part of the cell in your slice, and yeah, how do you deal with all of that? So that's the
yeah. So I think that they have their own challenges, but overall, they're both interesting.
Yeah, I think we're in an exciting time to be able to actually answer these questions. So it's going to be pretty cool the next few years.
Wonderful. Yeah. So any other questions? If not, then let's thank to the speaker again. Thank you, Dr. Scott. I think this was a fantastic presentation.
Amazing.
Thank you very much.
We will also post this online, and you'll have more people seeing your fantastic talk. And before we wrap up today, we'd like to also announce that next week, we have Dr. Daniel Dar from Weizmann Institute, and he used to be at Caltech. Actually, the project was about spatial genomics of bacteria, and he's going to talk about exposing the phenotypic structure of microbial populations via multiplex imaging. And with this, I think hope to see you guys next week. And next week, it's going to be at 2 p.m. in the regular time. So hope to see you guys at the regular time. And please enjoy your weekend, and thank you for your participation.
Thank you very much.
Thank you.
Thank you, Charlotte. Have a good weekend.
Have a nice weekend. Bye-bye.